Hello everyone, welcome back to our YouTube channel Pharmeasy. Today we will start the third unit of Human Anatomy and Physiology of B Pharm 1 Sem. This lecture will include introduction to blood, components, functions, hemopoiesis, hemoglobin and its formation, anemia, and the mechanism of blood coagulation. So, let's start. Blood. Blood is a connective tissue, and it contains plasma, platelets, red blood cells, and white blood cells, and it is circulated throughout the body. Blood supplies essential substances such as sugars and hormones to cells and organs and remove waste from cells. As the diagram shows, the red blood cells, which is also known as erythrocytes, white blood cells, also known as leukocytes, platelets, which is also known as thrombocytes, and plasma. Next topic is components of blood. It is divided into two parts, that is formed elements and the plasma. Formed elements are about 45% in concentration present in blood. These may be RBCs, WBCs, platelets. Plasma concentration in blood is about 55%. And it contains 10% nutrients and 90% of water. Now, this is a division of white blood cells. These are also known as leukocytes. These white blood cells can be divided into two categories, that is granulocytes and agranulocytes. Granulocyte is further divided into three categories, that is eosinophils, basophils, neutrophils. And a granulocyte is divided into two categories, that is lymphocyte, and the monocytes. Lymphocytes may be B lymphocytes, T lymphocytes, phagocytes. And the monocytes are macrophages and the dendrite cells. Our next and important topic is functions of blood. Blood provides oxygen to the cells as it absorbs oxygen from the lungs and transports it to the various body parts. Transport hormones and nutrients. It is a fluid connective tissue as it contains 55% of plasma and 45% of formed elements. Blood also helps in maintaining the internal body temperature by absorbing or releasing heat, which is known as homeostasis. Blood helps in the clotting of blood at the site of injury as it releases blood clotting factor. Blood transports the waste to the kidney and liver. As blood enters the kidney, where it is filtered to remove nitrogenous waste out of blood plasma. Blood also provides protection of body against pathogens. And pathogens are the disease-causing organisms. Next topic is hemopoiesis or hematopoiesis. It is the production of all of the cellular components of blood and blood plasma. The most common site of blood cell production is the spongy tissue inside our bone called bone marrow. And the hematopoiesis that occurs in the bone marrow is known as medullary hematopoiesis. Blood cells get made in our bone marrow and released in our bloodstream. There are various types of hemopoiesis, such as erythropoiesis, means production of RBC or red blood cell. Leukopoiesis, which means production of white blood cells, and the thrombopoiesis means platelet production. The lifespan or the survival period of red blood cells is about 120 days, and it is destroyed in spleen. White blood cells survive from a few hours to few days and the platelets survive for about 5 to 9 days. Now, look at this figure. Hematopoiesis that occurs in the liver is known as hepatic hematopoiesis. It is a feature of embryonic and liver of fetus, as hematopoiesis occurs in fetal liver and spleen. In fetus, after 20th week, the spleen fulfills the same function of hemopoiesis along with the liver. It continues to third trimester. After bone ossification, it occurs in the bone marrow. And stem cells are present in the bone marrow, which promotes formation of red blood cells, white blood cells, and platelets. Next topic is hemoglobin. Hemoglobin is a type of globular protein present in red blood cells. It is a respiratory pigment and helps in transporting oxygen as oxyhemoglobin from the lungs to different parts of the body. Hemoglobin present in human is coded by HbA1, HbA2, and HbB genes. The main function of hemoglobin is to carry and transport oxygen to various tissues. The hemoglobin molecule has 4 binding sites for oxygen molecules. That means each hemoglobin tetramer can bind 4 oxygen molecules. Next topic is formation of hemoglobin. There are various steps of formation of hemoglobin, as binding of succinyl coenzyme A that forms in the Krebs metabolic cycle with the glycine leads to the formation of a pyrrole molecule. 4 pyrrole molecules come together for the formation of protoporphyrin IX. And then, it combines with iron for the formation of heme molecule. Each of the heme molecule associates with a long polypeptide chain to form a subunit of hemoglobin referred to as hemoglobin chain. 4 of these chains bind loosely together, forming the complete hemoglobin molecule, as the figure shows. Next topic is anemia. It is a disease, disorder, or a condition that develops when blood produces a lower than normal amount of healthy red blood cells. Simply, it means lack of RBCs production in the blood. Anemia can be short-term or long-term. It can range from mild to severe condition. There are many forms of anemia, such as iron deficiency anemia, vitamin B12 deficiency anemia, aplastic anemia, and hemolytic anemia, and so on. Symptoms of anemia are pallid complexion, fast or irregular heartbeat, shortness of breath, chest pain, headache, and lightheadedness. Next topic is mechanism of coagulation. The mechanism of blood coagulation is based on hemostasis. This process includes 3 steps. First one is formation of prothrombin activator. Second is conversion of prothrombin to thrombin. And the third one is conversion of fibrinogen into fibrin. First is formation of prothrombin activator. In this, the formation of a prothrombin activator is the first step in the blood coagulation cascade of secondary hemostasis. It is done by 2 pathways, that is extrinsic pathway and intrinsic pathway. Second one is the conversion of prothrombin to thrombin. Prothrombin or factor II is a plasma protein, and it is the inactive form of the enzyme thrombin. And vitamin K is required for the synthesis of prothrombin in the liver. The prothrombin activator converts prothrombin to thrombin. Thrombin is a proteolytic enzyme. It also stimulates its own formation, that is the conversion of prothrombin to thrombin. It promotes the formation of a prothrombin activator by activating factor 8, 5 and 13. Last one is the conversion of fibrinogen into fibrin. Fibrinogen or factor 1 is converted to fibrin by thrombin. Fibrin threads are stabilized by the factor 13 or fibrin stabilizing factor. The fibrin stabilizing factor is activated by thrombin to form factor 13A. The activated fibrin stabilizing factor, that is 13A, forms cross-linking between fibrin threads in the presence of calcium ions and stabilizes the fibrin meshwork. The fibrin mesh traps the formed elements to form a solid mass called a clot. This is a simplified mechanism of coagulation. Prothromboplastine from the platelets in presence of calcium ion forms thromboplastin. Prothrombin in presence of thromboplastin and calcium ions forms thrombin. And fibrinogen in presence of thrombin is converted into fibrin. And fibrin forms fibrin polymers. Then, it forms a clot. I hope you like our lectures. And, if you have any queries regarding our lecture, then ask in the comments section. Thank you, keep watching, and take care.